This is a hematopoietic system physiology presented by DSHRTDB with a reference Cultural Strands of Physiology by Agamemnon Desopoulos and Stefan Silbernac. So, here we are going to visualize blood and its composition with all the physiology involved with it. So, now we are going to see the regulation of red blood cell. We start with the regulation of red blood cell production. We need to know that the main point involved in red blood cell production is going to be erythropoietin, and that erythropoietin is produced from the kidney and is going to be controlled by either a rise in partial pressure of oxygen, or you see that a rise in partial pressure of oxygen is going to cause inhibition of erythropoietin production, while a low partial pressure is going, or due to high altitude, is going to cause erythropoietin to be released. And now erythropoietin is going to move into the bone marrow and is going to stimulate the precursor cell conversion into erythrocytes, which are going to be released into the circulation. So now the next thing is hemolysis. You need to visualize what. The first point is that when you have red blood cells, when the red blood cells are being found inside the blood vessels, there's going to be high partial pressure of oxygen, and that high partial pressure of oxygen inhibits erythropoietin. But now when there is hemolysis, it also is going to cause a reduction in partial pressure of oxygen, and that reduction in partial pressure of oxygen causes stimulation of erythropoietin, which causes the conversion of precursor cells into the erythrocytes. Then the next point is going to be the life cycle of red blood cells. So life cycle, you need to know that it first starts from precursor cells inside the bone marrow. Then red blood cells are going to enter into circulation. So you need to know that the lifespan of normal red blood cells is going to be 120 days in a normal person. And now when it flows inside the blood, and inside the blood is going to break down. We have phagocytosis. You need to know that phagocytosis is when the cells are going to be waste up. So after that 120 days, so phagocytosis is going to occur extrasplenal. The extrasplenal phagocytosis is going to be inside the bone marrow, inside the lymph nodes, inside the spleen, and inside the liver. So these are all where the places where you have phagocytosis. So now the main position where you have phagocytosis is going to be the spleen. So when they are too old, they are going to be phagocytosed. And now you have the next part. This is the structure, the histologic field structure of the spleen. And now here we can see that there is a pulpal arteriole and there is a splenic pulp here. So these other cells here are the cells which are not going to have any problem, but when the cells are already worn out, they are going to be phagocytosed. So the next position that we are going to see are some parameters mostly involved with red blood cells. So now this parameter, the mean corpuscular hemoglobin, you have number one, the mean corpuscular hemoglobin. We have mean corpuscular volume. We have mean corpuscular hemoglobin concentration. So what is mean corpuscular hemoglobin? It refers to the mass of hemoglobin in all the red blood cells. The mean corpuscular hemoglobin, it refers to the average mass of hemoglobin in all the red blood cells. So if we take the normal, so you need to know that the normal red blood cell count inside the human body is going to range from 4 million to 8 million. The normal red blood cells inside the human body ranges from 4 million to 8 million, so even to 12 million. So these are 8 million normally. So now when all these red blood cells, you take all these red blood cells and you have the mass of each of the hemoglobin, the average mass of hemoglobin of these red blood cells is going to give the mean corpuscular hemoglobin. Now the next one is mean corpuscular volume. The mean corpuscular volume refers to the average volume of the red blood cells. So it refers to the average volume of a red blood cell. So you take all the red blood cell volume to divide by the total number of red blood cells that we have. And then the next one, you have the mean corpuscular hemoglobin concentration. The mean corpuscular hemoglobin concentration is similar or is equivalent to the hemoglobin level, but it's not actually equal to the hemoglobin level. So the definition of hemoglobin level, hemoglobin is defined as the hemoglobin present per unit blood. The mass of the hemoglobin level refers to the mass of the hemoglobin with respect to the volume of blood. While the mean corpuscular hemoglobin concentration instead means that it is the mass of hemoglobin, instead the mass of hemoglobin with respect to the volume of all the red blood cells which are found inside blood. That's the difference. For mean corpuscular hemoglobin, it is a mass of hemoglobin with respect to the volume of total red blood cells, while hemoglobin level is the mass of hemoglobin with respect to blood. That's why now the mean corpuscular hemoglobin concentration is going to be higher than the hemoglobin level, and this is because the total volume of blood is higher than the total volume of red blood cells, since red blood cells occupy 45% of blood. So now this formula, another formula of mean corpuscular hemoglobin is going to be hemoglobin concentration divided by the red blood cell count. Another formula for mean corpuscular volume is going to be the hematocrit divided by the red blood cell count. And another formula for mean corpuscular hemoglobin concentration is going to be hemoglobin concentration divided by hematocrit. And these are the values. For MCH, you're going to have 27 to 32 picograms. For MC, we have 80 to 100 femtoliters, and then for MCHC, we have 320 to 360 grams per liter. So now we continue now with the iron intake. We continue with the iron intake inside cells. So you need to know that iron is very important in the production of red blood cells. So iron intake is going to be very necessary. So this is the general diagram. So you need to know that normally iron intake is going to be, in females is going to be 10 to 20 milligrams per day, while in males is going to be 5 to 10 milligrams per day. So in females who can easily have children, they need 10 to 20 milligrams per day more than, because it can also need for the child. And in males we have 5 to 10 milligrams per day. So the iron, you have the iron here is going to, you see that this iron can enter as iron 3 iron. Now when it combines with hydrochloric acid inside the stomach, that hydrochloric acid is going to cause a reduction. Because of this hydrogen ion, is going to cause a reduction of the iron 3 ion into iron 2 ion. And now that iron 2 ion is capable of being reabsorbed. The non-ion which are being absorbed are normally 85 to 97% of what is being intake. But now let's see the fate of iron. So now inside the lumen we have iron 2 ion, we have iron 3 ion. So we need to know that what the ion which is being absorbed inside the body is going to be called iron 2 ion. So we have iron 2 ion being absorbed. So at this position iron 2 ion is being absorbed. So iron 3 ion is going to be converted by this FR protein. Is going to be converted first into iron 2 ion before iron 2 ion is going to be absorbed via the hydrogen iron 2 ion ATPase. And this hydrogen iron 2 ion ATPase now is going to cause iron to enter into the cell. Now iron which enter into a cell can bind to ferritin. So when iron 2 ion is inside the cell, it can bind to ferritin and is being stored. Else it can release iron iron 3 ion, and now the form of iron 3 of the form of iron which is being released out of the cell, out of the body and into the blood is going to be iron 3. So this iron 3 is the one that's going to be released when the cell is going to be turned over. And there's also going to be iron 3 here, mucosal transferrin. So we have iron 3 here, which is going to be in transferrin protein. So a transferrin proteins inside blood, you have iron 3 bounded. It binds to 2 ions. So now let's see iron storage. As I've initially said, you can have iron storage inside. You have 3 places where you have iron storage. In the first place, you can have inside the bone marrow. Second one, you can have inside the liver. Third one, you can have inside systemic blood. And the fourth one, you can have inside macrophages in the reticuloendothelial system. So we start inside the bone marrow. You see that iron inside transferrin is going to be stored in here, and the form in which iron is stored is iron 2 ion. So that's the form that iron exists inside a cell. Iron exists inside a cell as iron 2 ion, while iron exists outside the cell as iron 3 ion. That is a short note to put inside your memory. So inside this cell is going to be stored in ferritin as iron 2 ion. So, and is going to be used in the formation of hemoglobin. You need to know that hemoglobin contains iron 2 iron and not iron 3 iron. So the next point we are going to see is going to be liver. The liver, you have this iron 2 iron which enter, and then it can be used in the storage in ferritin, and that ferritin, another storage form can be hemosiderin. So that's another storage form of iron inside the liver. We have ferritin and we have hemosiderin. So the inside the systemic protein, we have inside the systemic blood, we have transferrin that can be used in the storage, but that's not actually storage because it's used mostly in the transfer. We have transferrin. We have the next one, we have hemopexin, and we have haptoglobulin. Haptoglobulin is when you need to know that anytime the cells are going to, the erythrocytes are going to break down, the erythrocytes are going to release hemoglobin inside the blood, and now that hemoglobin is going to bind with a protein called haptoglobin, and this haptoglobin is what is going to be involved in the transfer of hemoglobin throughout blood, because hemoglobin itself in blood is going to be toxic, so it needs to be bounded to something, and now that hemoglobin is going to be converted to heme after passing in the liver. It's going to be converted to heme, and this heme is bounded to hemopexin because heme itself is toxic to the body, and now heme can be reabsorbed by the macrophages. When it's reabsorbed by the macrophage, it's going to be producing iron, and that iron now is going to be stored in ferritin and hemosiderin, or it can be released back into the systemic blood and transferred by transferrin. So now the next point is folic acid and vitamin B12 cobalamin. So this is folic acid, 0.05 milligrams per day. For vitamin B12 is extremely very small, which is 0.001 milligram per day. So now we need to know that for vitamin B12 there's intrinsic factor which is being required, and this intrinsic factor is produced by the G cells of the stomach, and now these cells, now you see that the intrinsic factor, when it binds to vitamin B12, is capable of being reabsorbed at the distal part of the ileum, at the ileocecal junction. So now this could be reabsorbed by ATPases. So folic acid and vitamin B12 bounded to intrinsic factor are going to be reabsorbed by ATPases. Now you are going to have 7 milligram for folic acid and 1 milligram for vitamin B12, and now they are going to be stored. 7 milligrams is going to be stored for folic acid, and then 1 milligram is going to be stored for vitamin B12. Vitamin B12 is going to produce methylcobalamin, and this 7 milligram of folic acid is going to produce tetrahydrofolate. Now the tetrahydrofolate is going to N5 tetrahydrofolate is going to convert to tetrahydrofolate, and then it's going to enter into the folate regeneration. So this is time delayed. So we have time delayed at this position. We have dihydrofolate, and this dihydrofolate now going to convert into dihydrofolate reductase. So it's going to convert back to tetrahydrofolate, and here is another cycle. So this folic acid is going to be very necessary in the production of heme, so in the production of hemoglobin and all that. So this is what is very necessary. So we can now continue. This is DNA synthesis and all that is going to be required when you have erythrocyte production. So now the next point to put here, we are going to be the Forest-Lind-Vis effect. So all these are going to be, and they are going to be plasma protein. You need to know that the plasma protein inside the cell, the main proteins are going to be albumin. We have 60% of albumin. The total protein level, how 100% protein level, are 65 to 80 gram per liter. So we have 65 to 80 gram per liters of protein inside blood, 65 to 80 gram per liter of proteins inside blood. And now this for this 65 to 80 gram per liter, we have 60% is going to be only albumin, and then globulins are going to be the other 40%. Now of this 40%, alpha 1 globulins are going to be 4%, alpha 2 globulins are going to be 8%. So the total alpha globulins are going to be 12%, beta globulins are going to be 12%, and then gamma globulins are going to be 16%, and that's why it's going to be shown on this electrophoretic. It's going to be shown on this electrophoretic scheme. So where you place electrophoretic protein fraction, where you are going to place electricity into this and it's going to separate the different isoglobulins or isoproteins. So now we have these different part. We have now the different ions which are present inside the body fluid compartment, but we have already seen the body fluid compartment in one tutorial. So now we are going to see their different ion composition. We need to know that what a sodium ion we have inside the plasma is going to be 142 millimoles per liter. Inside serum, you need to know that serum is a portion that is going to be serum is plasma minus all the proteins. So now we see that the serum has a smaller volume, so the concentration in serum for all these ions is normally going to be higher. So sodium we have 142 in the plasma, serum 153, interstitium 145, cytosol that. Potassium is 4.3 inside the plasma, inside the cytosol, inside the cell is 140, and inside the cells for sodium is 12. So for free calcium, inside the plasma is 2.6, and inside the cell is going to be less than 0.001. The free magnesium is going to be 1 inside the plasma, serum 1, interstitium 0.1, and then you see it's going to be 1 inside the plasma, and then it's going to be 1.6 inside the cytosol. So now the next one, now these are going to be the sum of all these ions. Now we have anions. Chloride ion inside the plasma is 104, and then inside the cell is going to be 3. Hydrogen carbon is 24, and inside the cell is going to be 10. Inorganic phosphorus is going to 2, and inside the cell is going to be 30. Protein inside the plasma is 14, inside the cell is 54, and then the others we have 55.9, we have 5.9, and then we have 54. So all these are the concentration of the ions. Now we have the concentration of immunoglobulins. Now so this can be shown on this diagram. So you see here sodium ion is going to be higher outside the cell, then you have potassium, and then you have calcium and magnesium ion. Then at this position you have chloride ion, which is going to be the highest, then you have hydrogen carbon, and then you have protein and phosphate. For inside the cytosol, inside the cell instead, so at the level of interstitium of outside the cell, inside the cell you have potassium ion is going to be the highest, then you have sodium, and then you have calcium and magnesium. Here is protein is going to be the higher, then inorganic phosphate, then hydrogen carbonate inside the cell for anion. So now let's see the concentration of immunoglobulin in serum. So for the concentration, you need to know that immunoglobulin G are the highest. So you just follow the mnemonics GAMD in order to see the highest composition and then the smallest composition. So GAMD, so you need to know that one. Immunoglobulins G are going to be the highest concentration, and it's going to be 11 grams per liters inside blood. And then the next inside serum, then the next one we have A, which is immunoglobulin A for 2.25. In GAMD, A comes next, then M comes next, then D comes in, and then E comes last. So this is IG. G is going to be having 11 grams per liter, then A 2.25 gram per liter, then M 1.15, then D is going to be 0.03, and then E the last 0.0002. So that's going to be your simple mnemonics to remember the concentration from maximal to minimal concentration of immunoglobulins in blood. So now we need to know that what our immune system is going to be our acquired immune system, because you need to know that there is acquired and innate immune system. Innate immune system is what we have from birth, while acquired is what we have when we are exposed to a specific antigen inside the body. So now our acquired immune system is going to be divided into two main parts. You have humoral response and you have cellular response. Humoral response is mostly by the use of B lymphocyte, while cellular response is mostly by the use of T lymphocyte. So at this position here we have lysozymes. So in the humoral response you are going to have lysozymes, which are the necessary in damaging the cells. You have interferons. Interferon here we have interferon alpha, interferon beta, gamma are going to inhibit viral proliferation. You have interferon gamma activate macrophages, killer cells, and B and T cells. So we also have complement activation. All that is going to be complement activation, all that are going to act in the humoral response with all the antibodies. So in complement activation we have two main types of complement activation. We have the alternative activation. You have alternative complement activation, then you have classical complement activation. In alternative complement activation, there is going to be C3 production, complement factor 3 production, while in classical complement activation you have C1 cure activation. All of them are going to cause the production of C3b, and C3b is going to cause opsonization of the antigen. So C3 is going to bind on the antigen and cause now opsonization, make the antigen to be affinitive, to be to be attracted by macrophages. So this is what's going to happen. And we also have antigen antibody complexes. All of them are going to act with microorganisms. All of them add up with this classical alternative and classical complement activation plus antigen antibody complexes plus microorganism. All of them act to produce C3 that is going to produce opsonization. Also, another method of killing by the complement activation is that they are going to produce membrane attack complex. Membrane attack complex is when, in addition to this C3b, we have C5 to C9 that are going to add up to this C3b, producing a pore on the microorganism that is going to cause the release of material from the microorganism. Also, from the complement activation, we have C3a, we have C4a, and we have C5a. All of them are going to be necessary in the activation of the mast cells, basophil, the eosinophil, and also these are inflammatory factors that is, they are going to be used in the activation of mast cell, activation of basophil, activation of eosinophil, and activation of natural killer cells. So now let's see now the action of mast cells and basophils. So now when you have C3a, which are immune inflammatory factors, so all these are going to cause activation of mast cells and basophil. Now the mast cells act now by producing mediators of inflammations. So they can produce mediator of inflammation. Another way of activation of mast cell is when the antigen directly bind to the IgE antibodies which are found on the mast cells or the basophil cells. Mast cells are actually basophil cells, but mast cells are found inside the tissue and basophils are found inside blood. So now we have mediators of inflammation. So inflammation now occurs. So all this is going to cause inflammation. So that is for your humoral response. Then the next one, we also have activation of eosinophil. The activation of eosinophil occur when you have IgE that bind on the eosinophil, or when you have this complement factors that bind to eosinophil. So these eosinophil now are going to have different actions. So now we also have immunoglobulins which are being used at the level of humoral response. These are G, E, and MAD. So GAMD, all these immunoglobulins are involved here. These immunoglobulins have two portions. We have this FC portion, we have this FC terminal, and we have this other terminal. The FC terminal is going to bind with this FC terminal is going to bind with the cell of the body. This FC terminal is going to bind to a cell of the body, and this other terminal is going to bind with the antigen. So now in this position here, we see the membrane attack complex that is going to cause now sodium ion and water to enter, which is going to cause a burst of the cell. You can also have oxidants. You can have the release of perforins by the natural killer cells, because we initially said that natural killer cells are going to be activated by interferons, as you can see here. So we have interferon alpha, beta, gamma, which are going to inhibit viral progression, and interferon gamma also is going to be used in the activation of macrophages and natural killer cells. So now here you can see the action of natural killer cells by producing perforins, and these perforins now are going to cause the proteases, the perforation of the wall of the cells of this pathogen, and now there is going to cause the breakdown of the pathogen. Also, lysozyme action can occur, and this lysozyme action is from the action of macrophages. And we can also have the last action, which is going to be NADH oxidase, the oxidants produced by the phagocytes. So now we have at this position cellular immune response. For cellular immune response, we need to know that, so at this position we have antigen which are already produced, antigen which are already bounded to the antigens are already bounded to antibodies, or the antibodies here, or the C3b complement factor. So when it bounds here, binds like that, now you can have phagocytosis. So now phagocytosis is going to occur by releases. So we have oxidants are going to be produced by causing membrane damage. We have also proteases are all going to cause membrane damage, and we have mediators of inflammation, activated macrophages, and all that. So all that are involved in our acquired and innate immune response. So now we have specific immunity. So for specific immunity, like the T cell activation is also involved in the acquired immune response, in the cellular immune response. So now we have antigen presenting cells like macrophages, dendritic cells, and B cells. So these are antigen presenting cells. So when they present, they're going to present with the HLA1 or HLA2. So they present the antigen to the CD8 or the CD4 cells. So now when they present like that, there are other receptors that are going to bind for the action of this receptor to take place. So like the B7 receptor to the CD28, and the ECAM receptor on the LFA. So this is going to cause a dual signal, and this dual signal causes interleukin 2 to be released from the cell, and this interleukin 2 now is going to cause now the T cell proliferation. So now we have two main T cell proliferation. You can have CD8 T cell proliferation, and you can have CD4 T cell proliferation. CD8 T cell proliferation have HLA1, and CD4 have HLA2. So in this position for CD8, we have cytotoxic T cells. So cytotoxic T cells, we have infected tumor cell or foreign cell. What happened is that you have this ECAM, you have HLA1, you have CD94 from this position. All of them are going to respectively add up with the LFA1. We have CD8, we have T cell receptor, and we have the fast ligament. So when they bind, you have impulses. Impulses are now going to cause the stimulation of perforin of the vesicle containing perforin and granzyme to bind with the membrane of the T cell, and then be released on this tumor cell or infected cell. As now this perforin and granzyme are now going to cause apoptosis in this cell. So now in the next position we have CD4 and HLA2. So for CD4 and HLA2 we have T helper, or CD4 cell, also called helper cell. What occurs here is that the T helper cells, we have T helper 1, we have T helper 2. For T helper 1, activation here is going to cause macrophage activation. For T helper 1, we have activation causes macrophage activation, and then the release of interferon gamma here is going to cause inhibition of T helper 2. And also the release of interleukin 10 and tumor generating factor here, beta, is going to inhibit this T helper 1 cell. So these are macrophage activation at this position. So now we have specific immunity. Now the other one now is for B cell activation. So the B cell function, the activation occurs when these are naive B cells. Start with naive B cell. So what happens is that the antigens are going to bind to the different antibodies found on the wall of the B cells. These antibodies are immunoglobulins D, and can also be immunoglobulins M. So now this is going to be internalized inside the cell body, and when it's going to be internalized, you're going to have this HLA2 processing. And now the HLA2 processing now is going to cause now the binding of the antigen now with the HLA2, and then the HLA2 is going to be presented to the T helper 2. We need to know that T helper 2 functions in T helper 2 because we have two main CD4 cells. We have T helper 1 and we have T helper 2. T helper 2 functions in the humoral response. So it functions in humoral response, while T helper 1 function in instead in cellular response, where it's going to T helper 1 is going to activate the macrophages, and then the macrophages now are going to be very effective in phagocyting the cell, because it activates the macrophages to go and work more. While in T helper 1 is going to instead activate the T helper 1 instead T helper 2 instead activate the B cells. So you see that the B cell come and present the antigen with the HLA2, and when they present with HLA2, what happens now is that we have impulses transmission. So interleukin 4, interleukin 5, interleukin 6 are produced by the T helper 2 to cause now activation of the naive B cell to do proliferation. Now the naive B cells are going to grow up to form mature B cells called plasma cells, which are going to produce different series of antibodies like immunoglobulins G, A, M, and E. So specific humoral response. So all that. So now the next one, we have anaphylaxis. So what occur in anaphylaxis? In anaphylaxis, we have immunoglobulins G. Immunoglobulins G are first going to be produced during the first anaphylactic reaction. So in anaphylaxis, you need to know that we have different stages of anaphylactic reaction. The first stage is that we have the first contact with the antigen. So the first contact with antigen, you see that in this case there are not going to be any response. So when you have the first contact, the antigen is going to propagate inside your blood, and then it's going to be uptaken by the B cells. When the B cells uptake the antigen, they are now going to process the antigen and produce corresponding immunoglobulins for these antigens. Now the corresponding immunoglobulins produced are called immunoglobulins E, and now these immunoglobulins E are now going to move all through blood, and when they move all through blood now, is going to bind to receptors on basophils. These receptors here is going to bind to receptor IgE receptors on basophil. So now you have IgE receptors now are going to be permanently bounded on basophils and even mast cells. Now you have antigens. Now when you have a further contact again with that antigen, example a PET, what happens is that the antigen is going to bind to now the IgE on the mast cell, and that's going to cause now activation of that mast cell, and during that second contact now you have an inflammatory response called anaphylaxis. It's going to be due to the release of histamine, the release of PAF, the release of leukotriene, and other inflammatory mediator. So now serum sickness. For serum sickness, you can look for the antigen, you can look for the antigen antibody complexes, you can look for the symptoms, and you can look for free antibodies. So you have day after the first antigen exposure. So you need to know that to know for serum sickness. So when it is still the first days during exposure, the concentration of antigen is going to be high, extremely high. Now at the middle, between 5, so when the first 5 days, you see that the concentration of antigen is going to be extremely large. Then between 5 to 10 days, you have concentration of antibody antigen antibody complex. So when you have antigen antibody complex, we have now this antigen when you have antigen antibody complex being formed, you have symptoms at this position here. And now the more the antigen reduces, the more the symptom regresses. So here you can visualize the symptoms of that disease when you have antigen antibody complex and when you have the antigen. Now when you see free antibodies inside the blood, it implies that the symptoms are going to be less inside the body. That is, the body is much more eradicating the disease. Then we have the ABO blood group compatibility. We need to know that there are 4 main blood groups. We have blood group O, meaning that there is no A nor B there is no A nor B hemolyzing, that is anti-A or anti-B agglutinins. So they are not there. So in blood group A, you instead have an anti-B hemolyzing or an anti-B agglutinin. So if you place blood group B in this person, or blood group AB if you place blood group B or blood group AB in this person, they are going to agglutinate. So the next one, we have if you place blood group B in this person, because the blood group B is going to contain if you place blood group so this is A. So if you place blood group B, because blood group B contains B, so that's going to be a problem. So it's incompatible with blood group B in this position. It's both incompatible to blood group B and blood group AB because AB also contain the B antigen because it has the anti-B agglutinins. Now for blood group B is going to be incompatible. For blood group B is going to be incompatible. For blood group B is going to be incompatible to both blood group A and blood group AB. So it's both going to be incompatible with blood group A and blood group AB. So now the next one we have AB now is going to be incompatible to, is not going to be incompatible to any blood. So now we have the next one now. So blood group, that's why blood group O is the universal donor. The universal donor because there is no antigen, so there is no reject. While blood group AB is a universal acceptor because there is no agglutinins inside the blood. The recipient with blood group AB, and thus it can accept any antigen. So now we have also resosensitization. For resosensitization, what occurs is that if a mother has a first child who is rhesus positive and the mother is rhesus negative, what occurs is that they are first going to have IgM production. So when the child is going to come out, there's going to be a fetal-maternal blood transfusion. After that fetal-maternal blood transfusion, blood is going to enter into the mother and going to cause stimulation of the cells of the body. Now that humoral stimulation is mostly going to produce IgM, and now the IgM are very large and thus they cannot traverse the placenta. So since it cannot traverse the placenta, the first child usually comes out unaffected, but the second child is going to have the problem. If the mother say have a rhesus positive child and then the mother is rhesus negative, the second child is going to have severe hemolysis and may also die, because in this case you have when there's going to be a conversion of IgM to IgG, and now the IgG now is going to traverse the placenta because it's smaller, and now it's going to damage, cause severe hemolysis in the child. So now these are the different points. So now the next one, we have platelet mediated hemostasis. So now we first need to know that when you have an endothelial damage, the platelets are going to adhere on the wall of the cell. As you can see here, you have collagen at this position here. The platelets are going to adhere with the von Willebrand factor. Both of them are going to adhere. So you can see here, this is platelet adhesion. So it first starts with, at this position here, you have the platelet, you have platelet addition at this position. You can also have platelet activation. So now the first thing is platelet adhesion that occurs with the von Willebrand factor and the collagen, which is found below, and the von Willebrand factor, which are found at the subendothelial level. Now when it binds to the von Willebrand factor and the collagen at the subendothelial level, now that's going to cause now platelet activation. Platelet activation is going to cause a secretion of serotonin, thromboxane A2, ADP, and all that. Now serotonin and thromboxane A2 are both going to cause vasoconstriction, which is going to cause slower blood flow and more platelet activation and adhesion. And von Willebrand factor is mostly involved in a platelet adhesion. It's going to also be secreted from the platelet after platelet activation. And lastly, you have platelet aggregation and clot formation. So now when the clot formation occur at the last portion when the platelet have already occurred. So now how does clot formation, blood clotting occur? Blood clotting occur in two main ways. We have the exogenous activation, or we have the internal activation. We have exogenous and we have endogenous activation. So this is tissue injury, so in contact with the tissue, and this is due to in contact with the blood, endogenous. So now contact with the collagen, so and all that. So now what happened, and this is all you can still be called the intrinsic and the extrinsic pathway. So here is the extrinsic pathway, and here is the intrinsic pathway. So now the extrinsic pathway, we see that it first starts with clotting factor 7, The clotting factor 7 is going to bind with clotting factor 3, that is calcium, to produce activated clotting factor 7. And now activated clotting factor 7 is now going to cause a conversion of clotting factor 10 to activated clotting factor 10. Now in the intrinsic pathway, what happens is that we have clotting factor, start at clotting factor 12, converting to clotting factor 12, activated clotting factor 12, and now this is because of kallikrein and other kallikrein bodies. Now activated clotting factor 12 is now going to cause the conversion of clotting factor 11 into activated clotting factor 11. Activated clotting factor 11 causes the conversion of clotting factor 9 into activated clotting factor 9, and activated clotting factor 9 binds now with clotting factor 8 and now clotting factor 3 to convert the activated clotting factor and to convert clotting factor 10 into activated clotting factor 10. And now activated clotting factor 10 now can act directly on, you can bind now with clotting factor 5 and act directly on prothrombin, and prothrombin is going to be converted to thrombin, which is activated clotting factor 2, and that thrombin now is going to cause now the conversion of fibrinogen now, which is clotting factor 1, to activated clotting factor 1, fibrin, which is going to produce now fibrin complex, and now fibrin is going to produce a meshwork that is going to cause this binding aggregation as such here and produce your blood clot. So now this is fibrinolysis. So fibrinolysis, we have different proteins that can be used in fibrinolysis, and this we have the first one, we have plasma kallikrein. So kallikrein we can bind due to clotting factor 12. Kallikrein binds plasminogen. We have plasminogen, and which is converting to plasmin. Plasmin is a main fibrinolytic portion. So that plasminogen converting to plasmin, that plasmin is going to cause now the breakdown of fibrin meshwork to soluble fibrinopeptides. So it first start, the activation of this plasminogen to plasmin occurs with plasma kallikrein. So activation of plasma kallikrein to kallikrein is going to occur via clotting factor 12. So this kallikrein now is now going to activate plasminogen. The second thing is urokinase. Urokinase also activate plasminogen. The third thing is TPA also activate plasminogen. We also have streptokinase called from streptococcus, and we have staphylococcus from staphylococcus. So all these are anticoagulants. So all convert in plasmin into plasminogen to plasmin. The next one, which inhibit conversion of plasminogen to plasmin, we have aprotinin, and the inhibition of plasmin to cause the conversion of fibrin meshwork into soluble fibrin. We have aprotinin, we have tranexamic acid, we have tranexamic acid, we have alpha antiplasmin. So now let's see now suppression of coagulation. The suppression of coagulation mainly occurs via antithrombin 3. So antithrombin 3. Now we have the different activation of antithrombin 3. So antithrombin 3 is going to cause the inactivation of exogenous cycle, that is the extrinsic cycle. Inactivation of the intrinsic cycle is also going to cause inactivation of conversion of the prothrombin to thrombin, and the inactivation of action of thrombin to production of fibrin. So now let's see now what is going to activate the antithrombin 3. We have heparin. Heparin is going to activate antithrombin 3. That is going to cause the inhibition of the exogenous cycle, that is the binding of clotting factor 3 to clotting factor 7. Is also going to activate the inhibition of the clotting factor 12 activated to produce clotting factor 9, and also prevent activated clotting factor 9 to produce clotting factor 10 at the level activated clotting factor 10. So the next one, we have heparin binding to antithrombin, preventing the action of the complex of activated clotting factor 9 and activated clotting factor 8 to act on the clotting factor 10 to activated clotting factor 10. So you have this. It also prevent the action of activated clotting factor 10 on prothrombin, and also prevent the action of the thrombin to fibrin network. So all these are functions. So from here we say thanks for your kind attention, and please don't forget to like and subscribe for the channel. Thank you.